Good morning, everyone. Today we are going to finish the remaining topics from the skeletal muscle. So, first, we are going to start with the excitation, contraction, coupling. To understand the concept of this excitation, contraction, coupling, we need to start with the understanding of a structure which is specialized in the skeletal muscle, and it is the sarcotubular system. This system, this sarcotubular system, comprises of two physiologically and structurally tubules, which is known as the T tubule and L tubule. T tubule means transverse tubule and L tubule means longitudinal tubule. If we are to draw this in a simplified way, it stands out to be like this T tubule. This is the sarcolemma. This T tubules are nothing but the transverse invagination from the sarcolemma. Sarcolemma is the plasma membrane of the skeletal muscle, as we know. So, the T tubules, they are the finger-like projection that has gone inside, that has invaginated from the sarcolemma. This is the sarcolemma or the plasma membrane of the skeletal muscle fiber, and these are the T tubules, transverse tubules. Now, on these T tubules, there are this characteristic presence of this voltage gated calcium channel, which is dihydropyridine receptor. On the either side, on the either side of this T tubule lies the longitudinal structure, like which is lying longitudinally, like this. Okay, longitudinally, like this. Now, these tubules, which are longitudinally oriented, these are called as the longitudinal or L tubules. The characteristics of this L tubules are that they also have a calcium channel. So this calcium channel that they have here, they lie something like this. And these are called the ryanodine receptors. These are called the ryanodine receptors. This longitudinal tubules, this is a part of a Solon structure. Like this actually lies as a like a Solon structure, like this on the either side of the transverse tubule. Okay, it's like a Solon structure. So if you look at it, there is a segment or a portion of the T tubule, and on either side lies a portion of the T tubule, this portion, and on either side there lies two Solon structure. Now this Solon structure is called as terminal cisternae. Now T tubules plus one terminal cisternae linked to L tubule, and then another T tubule, they make up a structure which is called as the triad, So in a triad of a skeletal muscle, there are two tubules and one L tubule, that is a part attached to the terminal cisternae. Now, this is very important for the excitation contraction to happen. Here, in normal condition, drawing the structure again in a little bit simplified manner, like in normal condition, this is a T tubule and this is our L tubule. This is T tubule and these are the L tubules. Now, in resting condition, a gap of 10 nanometer exists between the T and the L tubules. Okay, there is a gap of 10 nanometer between the T tubule and the L tubule. That means during resting condition, there is no physical contact between the T tubule and the L tubule. It is only during the contraction that the dihydropyridine receptor and the ryanodine receptor has physical contact. Like they come so closer to each other that the gap of 10 nanometer is no more. Therefore, what is exactly excitation, contraction, coupling? Excitation, contraction, coupling means the release of calcium through the voltage gated calcium channel. Now, what is the voltage gated calcium channel? That is our dihydropyridine receptor that opens the non-voltage gated calcium channel, that is the ryanodine receptor. So first there is release of the calcium through the voltage gated calcium receptor, then it induces a confirmatory change in the ryanodine receptor, which is a non-voltage gated calcium channel, and then there is this physical contract between the two, which leads to the contraction of the skeletal muscle. So this lies something like this. This is the cytosol, and this is the sarcoplasmic reticulum. Upon excitation, say this is the dihydropyridine receptor, the calcium enters into the cytosol, which causes the opening of the ryanodine receptor, which is a non-voltage gated calcium channel, and this also pulls out calcium from the sarcoplasmic reticulum, and in doing so the calcium induces excitation, and there is physical contact between the dihydropyridine as well as the ryanodine receptor. Okay, now this is the excitation contraction coupling. This ultimately leads to the this leads to the contraction of skeletal muscle. Contraction of skeletal muscle means contraction of the or contractile changes that happens in the skeletal muscle fiber. So, in the process of contraction of the skeletal muscle, there are two main parts. First part is the contraction part, and second part, it is the relaxation part. Okay. Now, in the contraction part of a skeletal muscle, there are further like subdivision, like there are certain events that are helping in the contraction of the skeletal muscle. So we will study this event wise supported by the diagram. The first event, the events that are happening, the first event is release of calcium, release of calcium. Now this release of calcium leads to the second part, that is lifting of inhibition from actin molecules. Third part is powering of the myosin. Now, actin inhibition lifting and myosin powering, these two happen like side by side. Okay. Then, physical interaction and power stroke by the myosin. Okay. Event wise, there are thus 4 events that is actually happening. Okay. Now, this is your contraction part. Going to the relaxation part, the event that happens in the relaxation part is like unengaging or unbinding of the actin and myosin molecule. Okay, these are the events that is happening in the process of contraction and relaxation. So going by this event, we will study one by one the events that are happening. So to start with the contraction part. The contraction is best explained by the sliding filament theory. Okay, it is best explained by the sliding filament theory. Going by the event, first is release of calcium. Now, how if we go back to the neuromuscular junction that we studied, neuromuscular junction, it is a specialized junction through which a motor spinal neuron goes and innervates the skeletal muscle, right? That is what is a neuromuscular junction is. Okay. Now, release of calcium happens how? It happens like first action potential reaches the post synaptic membrane. This mechanism we know, we have studied, so I'm not going into it again. So once the action potential reaches the post-synaptic membrane, after the acetylcholine has bound on the acetylcholine receptor and there has been wave of depolarization generated, then what will happen? Then the calcium enters the cytosol via the dihydropyridine receptor, which are the voltage gated calcium channels. Now, this induces conformational change in the ryanodine receptor, and as soon as this conformational change is induced, what happens? There is more release of calcium from the sarcoplasmic reticulum to the cytosol via the ryanodine receptors. Okay, this is how the calcium is released in the sarcoplasm from the sarcoplasmic reticulum. Okay, next step is the lifting of inhibition from the actin molecule. Okay, so last class, what did we study in the last discussion? We saw that if this is just taking one actin molecule for our better understanding, if this is the actin molecule and let's say this is the active site on the actin molecule through which it will bind to the myosin. Say this is the active site, that is a myosin binding site. Now this one is closed by or covered by the this is troponin molecule with its I inhibitory unit, and on top of that, here this is the troponin molecule inhibitory unit, and here on top of that we have the C subunit of the troponin, and there is the T subunit of the troponin through which it is bound to the tropomyosin. Now the calcium that has entered, that has gotten released, calcium that is released into the sarcoplasm binds with the C subunit of troponin. So something like this happens. Like this is the actin with its active sites, okay, and this is the troponin with the I. Okay, so here in the C unit, in the C subunit of the troponin, calcium goes and sits on it. Calcium goes and sits on it. Now, as soon as calcium goes and sits on it, there is a conformational change in the whole arrangement. Like this is the actin molecule with a binding site, and this is the troponin, and once the calcium is bound, it sways, it pulls the inhibitory site like this, it pulls the inhibitory site that was bound onto the actin molecule, and it exposes the binding site on the actin. So now you see the binding site is the actin binding site is exposed. The actin binding site is exposed. So this is the second part, that is activation or lifting of the inhibitory subunit from the actin molecule. Now next is the activation of myosin. How is myosin activated? In normal condition, so this is our myosin. For our understanding purpose, we are drawing it. Say this is the myosin head. Okay. So in resting condition, like when there is no contraction, this myosin head, it will have one ATP bound to it. This is the myosin head, the globular head, and this is the body with the neck and all. And this is a heavy chain. So thick chain. So this has a ATP bound to it. Interesting. The ATP bound to myosin is hydrolyzed. We know that in the myosin, what do we have? We have one ATP binding site, we have one catalytic site, right? So here on this ATP, the myosin that is bound, the ATP that is bound to the ATP, it will be hydrolyzed by ATPase. So this will be hydrolyzed by ATPase enzyme. So once it is hydrolyzed, then obviously the ATP molecule that is there, it will break into ADP and IP. Once ADP plus IP is attached, then the myosin is empowered. Means it is ready to bind. Okay. Comes the third event, the actual binding. Is the here, like just for us to remember, like release of the calcium we have studied, lifting of the inhibition, empowering of the myosin we have studied. Okay, now we will study the actual contraction or the sliding filament theory. So here what happens is, like, we will study it with a diagram. Okay, now if we look at the picture, this is the amalgamation of all the events that we have discussed. So first, in the relaxed state, we can see the myosin is like not powered and it has got the ATP bound to it. In this condition, the myosin is not at all capable of causing any contraction. After the hydrolysis of ATP, it breaks down into ADP and IP, which is the energized or the empowered form. But in this condition also, the ATP has not yet bound to the myosin, and that is the reason why myosin is now empowered. In this condition, at a degree of 90 degree is maintained and it approaches to the myosin. As you can see here, the myosin head is now heading towards the actin at a degree of 90 degrees. Now, this approach and the close distance, very close distance they lie. This myosin and actin lie in this condition, and the complex, the whole myosin and actin, which is so close to each other and has just started the physical contact, in this condition, it is called the actomyosin complex. Okay. Now, in this actomyosin complex, there is bending of this myosin. As you can see, the myosin through the flexible neck region, it has formed a 45 degrees. It has formed a 45 degrees and it has touched on the active site of the actin. Now, as soon as it touches the active site of the actin, next what happens is release of the ADP and IP, release of the ADP and IP. And once the ADP and IP is released, it gives the power stroke. This is the stage where we see here we see the power stroke. Now, important point to remember is that power stroke will not happen until the ADP and IP is dissociated from the myosin, and this is what is the sliding filament theory, because in this condition you can actually see the actin is kind of sliding. It is sliding on the myosin. It is sliding on the myosin. So this configuration is called the sliding filament theory. Okay, let us just have a repeat of it. So first, what did we study? First is the activation of the actin, which is not shown here, but I have showed you in the diagram. So here, let's assume the actin has been activated. Okay. Now, myosin in the relaxed condition, how does myosin stay? Myosin is attached to ATP. When ATP is attached to the myosin is powerless. It cannot undergo binding. It cannot bind with actin. So, myosin and ATP, no binding with actin. Okay. The hydrolytic site. In the hydrolytic site, this ATPase enzyme breaks down the ATP into ADP and IP. So once this ADP and inorganic phosphate IP is formed, it is activated, it is energized. In this energized condition, it lies at a 90 degree in respect to what? 90 degree in respect to the actin molecule. Now, in this 90 degree form itself, it moves closer towards the actin molecule, which is having now the binding site open. So when this myosin moves closer to this actin, what is this configuration called? This configuration is termed as actomyosin complex. Acto from actin, myosin from myosin. So this actin and myosin, which has minimal gap at a 90 degrees now, is called as actomyosin complex. Now, after this has happened, immediately there is like bending. We have studied that the neck of the myosin is flexible, it can move, it can bend. So this neck kind of bends, and the actin binding site on the myosin, it goes and touches the actin. It goes and touches the actin by bending its neck. So it was 90 degrees, and when it is touching the actin molecule, it bends and becomes 45 degrees. Now when it is at 45 degree, it pushes onto the binding site on the actin. This is called as the power stroke. This is called as the power stroke by the myosin on the actin. This is what causes the contraction of the muscle. But just before the power stroke, what important event happens? The important event that happens is that the ADP and the inorganic phosphate, the ADP and the IP, inorganic phosphate, both of them gets dissociated, just moves away from the myosin head, because only when they are dissociated the power stroke can be completed fully. Okay. And the orientation, the physical orientation in which this actin and myosin is lying, is kind of actin molecule is sliding over the myosin molecule. That's why it is called as the sliding filament theory. It is called as the sliding filament theory. It is sliding filament orientation because during power stroke, the actin is sliding over the myosin. So that's why it is called as the sliding filaments orientation, and the theory that describes it is the sliding filament theory. Now, one thing, this attachment after during the power stroke, when the myosin is binding to the actin, it is very rigid. It is very, very rigid. And there is nothing in this condition that can break away the myosin from the actin. That is, the contractile state of the skeletal muscle cannot be broken down, cannot be ended as long as the myosin is bound to the actin in this condition. So, for remembering, we write down as long as the myosin head without ADP and IP is attached to the actin, nothing can cause the detachment. Okay, this is the contraction or the contractile part of the skeletal muscle. This is how the contraction happens. The second part, that is the relaxation part. Okay, how does the relaxation part happens? For unbinding of myosin from actin in its rigid state, and ATP and ATP molecule has to bind again with the myosin head. Okay, this again, let us have a better understanding with the diagram. Okay, okay, so let's have a look at this picture. Okay, so this is the same, this is the event, same event that is depicted in a different way. So here we can see this is a energized myosin head that has bound at 90 degrees, and after that this is the power stroke and the release of this ATP and IP. Okay, this is a stroke which causes a contraction. Now this is the most rigid part. This is like, in this part there is nothing that can dissociate it. It is the rigid complex. Now contraction is over, relaxation has to happen. Now for this relaxation or the unbinding of the myosin from the actin will again happen when this ATP goes and joins the myosin head. When it goes and joins the myosin head. So once another molecule of ATP goes and binds with the myosin, see here the detachment. Here the detachment, the detachment of the myosin happen from the actin. The ATP kind of pulls the myosin away from the actin. Okay, this is how the myosin is again brought back to low energy state, because when ATP is bound to myosin, it is a low energy state. When ATP breaks down and ATP also dissociates, it is a highest energy state. So like that, the relaxation happens, again the next cycle resumes. So every time, so for every contraction and relaxation to happen, ATP is essential. It is a must. Without ATP, contraction and relaxation cannot happen. Okay, especially the relaxation cannot happen. One additional point to keep in head is that during the power stroke, the myosin molecule pulls the actin tail part by towards itself by 11 to 12 nanometer. It pulls towards itself by 11 to 12 by nanometer. And we have seen the mechanism is true, because what we have studied in the sarcomere also explains supports the same finding, same theory. Okay. Now, the applied part of it. The applied aspect of contraction and relaxation of the skeletal muscle. What is the applied part of it? The applied part of it is what we see in rigor mortis. Now, what is rigor mortis? Okay. Now, this is also called as cadaveric rigidity. Okay. It is also called as cadaveric rigidity. Now, what is this cadaveric rigidity or the rigor mortis, and why does it happen and how does it happen? After death, whenever a person dies, what do we see? We see the body of that person becomes hard, becomes stiff, right? So after death, within 12 hours, there is stiffening of the body. So why does the stiffening happen? What is the reason behind it? The reason is that after death, the metabolism stops, right? The metabolism stops. No metabolism is happening. No ATP is generated. There is no generation of ATP. ATP stock gets over. Okay. Once the ATP stocks get over, then what will happen? We have seen for the relaxation to happen, one ATP molecule has to bind with the myosin molecule. No, thus no ATP molecule to bind with myosin. So muscles were always already in contracted stage after death, during death. So there is no ATP that can bind with the myosin head, myosin molecule. So what happens is that no ATP molecule to bind with the myosin. Relaxation or unbinding of myosin from actin not happen. So there is like this rigid state of the muscle as the myosin is not getting attached from the actin. Now the order in which this rigidity happens is that as per Newton's rule, rigidity post death starts with the muscles of mastication, muscles of the face, mastication muscles, then face and neck, then trunk and upper limb, and last to the legs. So the stiffening starts with the mastication muscle, obviously the face, the neck, the trunk, then the arms, and at last the legs. Now this rigor mortis, it does not stay forever and ever. It stays only for like 12 to 14 hours max. After about 12 hours, the passing off or reverse, like loosening up, the relaxation, kind of like the loosening up, reverse state happens in the same sequence. In the same sequence. Like the loosening up also starts with the muscle of the mastication, face, neck, trunk, upper arm, and at last the legs. Okay, so this was our contraction relaxation with the rigor mortis, the applied part in details. Okay, now we will move on to the next part, which is the motor unit. So, what exactly is a motor unit? The motor unit, it comprises of certain parts. Like it comprises of ventral horn cell, right? It comprises of ventral horn cell. It comprises of a spinal motor neuron. It comprises of skeletal muscle fibers. Okay, so drawing the motor unit, it is like this. This is our ventral horn cell, the dorsal and the ventral. So here, if we draw the spinomotor neuron that is coming from the ventral horn cell, it will be like this. And these are innervating like the skeletal muscle fiber. Various skeletal muscle fiber. Like this. If these are the skeletal muscle fiber, so they are innervating over here like this, branched out and innervating. Okay, so a motor unit is the motor nerve fiber that innervates skeletal muscle fiber. Now, one motor nerve may innervate 3 to 6 muscle fibers. Okay. Example, eyes and hand, like ocular muscle and the muscle of the hand. Or one motor nerve may innervate 500 to 600 muscle fibers. Example, muscles of the back. Okay. So, motor unit means one single motor neuron, alpha motor neuron, that is going and innervating how much ever skeletal muscle fiber it can. So each one is a motor unit. Now, what are the characteristics of this motor unit? What are the features or what are the characteristics? The characteristics are they follow all or none law. They follow all or none law. They contract synchronously. Together they will contract. Okay. And they follow the size theory. Now, follow the size theory. The size theory is also called as the Henneman theory. Okay, it is also called as the Henneman theory. What is this Henneman theory? This size theory or Henneman theory, or we can also say theory or principle. What does it state? Henneman theory or this size principle, what does it say? It says that it states that smaller motor units. Smaller motor units mean when one nerve fiber is innervating lesser number of skeletal muscle fiber. So it states that smaller motor units are recruited first. Means they they get the wave of contraction faster, sooner. Okay, first, and then the larger motor units get recruited. Means motor unit that are having more number of muscle fiber, they kind of contract late. This is one. And they follow the all or none law. Either upon contraction, either the whole motor unit, like all the skeletal muscle fiber that particular motor nerve is innervating, that will contract or it will not contract. It's not that like out of this 500 skeletal muscle fiber, 200 will contract, other 2 or 300 won't. No, this will not happen. The whole of motor unit will contract upon receiving a stimulus or they will not contract at all. This motor unit recruitment increases with the strength of stimulus. What does that mean? What does that mean? Say this is the number of motor units. Okay, and this is like the stimulus has been applied, and this is like the contraction of motor units. So on the lower stimulus, when the stimulus is like less strong, it will be seen that only some motor units are, like the smaller motor units are contracted, are recruited. The others are not contracted. Now, if we increase the stimulus, when we increase the stimulus, then there will be gradual increase in the contractility, like that. We further increase, we get more. We further increase, we get more. So like that, as long as the strength and the intensity of the stimulus is increased, the number of motor unit that is recruited increases. This is another characteristics. Now we are talking about this contraction and relaxation, right? One term that needs to be remembered is the twitch. So what is a twitch? Twitch, it means one contraction and one one contraction and one relaxation. Okay, so drawing this, if this is the curve, this is the tension generated, and this is the stimulus induced contraction. So if the stimulus is induced here to a muscle fiber, what we see is a twitch. The twitch is something like this. The twitch is something like this. So this is like the phase of contraction and then phase of relaxation. That is a twitch. Now this is contraction of a muscle fiber in response to one twitch. Now if there are multiple twitch or two twitch like given, which has resulted from two simultaneous stimulus, say this is one stimulus and here two stimulus are given and here multiple stimulus are given back to back. Then what do we see? In response to one stimulus, we get a single twitch. In response to two stimulus, then it may sum it and it may give a two twitch, or it can be in a declining mode also, decremental twitch also. Decremental twitch means initially the first half may be bigger and then the second half may be smaller. Now, if a multiple number, like numerous number back to back stimulus is given, then it might result in a non-responsive phase for a long period of time, which is called as a tetanus. So, this is a single twitch in response to a single stimulus. This is a summated wave in a skeletal muscle fiber when two consecutive stimulus is given back to back and the second one is in a relative refractory period, and this one is a tetanus in which numerous number of stimulus is given back to back constantly without stop. So once the stimulus is stopped, then eventually there is decline in the contraction. But in between there will be this plateau state, means the muscle has contracted and it has not shown any more contraction or relaxation with further stimulus. So when repeated stimulus is given to the skeletal muscle, what happens is that it goes to a state of rigidity as long as the stimulus is staying, causing it very tight. This is called a tetanus, the rigid state when there is numerous number of stimulus given back to back. So these are the various type of waves of contraction that one can see in a contracted muscle fiber after this contraction stimulus has been given. Okay. Coming to the types of skeletal muscle fiber. The skeletal muscle fiber is of more than one type. It is classified into skeletal muscle fiber or fibers classified into type 1 fiber and type 2 fiber. Now this type 1 fiber is also called as the slow type of fiber, and the type 2 fiber is called as the fast type of fiber. Okay. Now this this type 2 type of this muscle fiber, it is further divided into two more types. One is the type 2A and type 2B. Now, type 2A is also called as the fast fatigue resistant type 2A, whereas the type 2B is called as the fast fatigable fibers. Okay. This two type of fibers, now if we take the subdivision of the type 2 also, it is like three type of skeletal muscle fibers. They have distinctive structural as well as functional characteristics which are quite similar and also not so similar from each other. Studying those, they are like the type 1 muscle fiber, type 2A and type 2B. Okay. What are the characteristics feature of them? First, we will go with the synonyms of it. Like type 1 is also called as slow oxidative type of the muscle. This is called as the type 2A fast glycolytic, and this is also fast glycolytic. Okay. Characteristic color. The first characteristic is the color. How do they look? How do they look color-wise? Okay. This type 1, it looks red in color. Whereas this type 2, they are like white and white. Okay. Second is the capillaries and mitochondria. The capillaries, they are high. They are like in large number. This is lesser and high, less and lesser are like in comparison to each other. So type 2 is lesser than the type 1 and then type 2b is the least. Okay, mitochondria, it has in largest quantity, moderate quantity and in small quantity. That's why this type 1 is red in color because of presence of this capillaries in mitochondria, and the other one are the not so red in color. White doesn't mean like it's milky white, but it is like much whiter than the type 1 type of the skeletal muscle fiber. Okay. The calcium pumping capacity. Now the calcium pumping capacity, it is moderate. Okay. And here this two, they have high capacity. Next is the myosin activity. Here the myosin activity is relatively quite high, but in other two it is small activity, small activity. The diameter of this skeletal muscle fiber. The diameter of the skeletal muscle fiber, it is small. It is small in diameter. It is intermediate, means compared to the type 1 and type 2b, and the type 2b, it is like big or the largest one. Okay. In terms of recruitment, when action potential come in terms of recruitment. Okay. It is recruited first because it is a small type, and we know from the size principle theory that the small skeletal muscle type, they will get recruited first. This is like second, and this will be recruited last. The oxidative capacity. Oxidative capacity is high, whereas it is low and low here. The glycolytic capacity. Now here it is least, here it is moderate to high, and here it is high. Okay. Now location. The location of the muscles. Now this is located muscles that maintain posture. Okay. Muscles, here the type 1A, 2A muscles that are used while running. Your posture means during walking mainly. And where this type 2B are located, these are located in the muscles that are used during sprinting. Okay, so these are the characteristics type of the skeletal muscle fiber. Now, moving on to the next section, which is the sources of energy. Now, sources of energy for the contraction of muscle. We know that for contraction of muscle and also the relaxation part of it, ATP is an essential factor. The currency of energy is ATP. So what are the various sources from which this ATP reaches to the muscle fiber? First, obviously, it is the ATP store. Okay, so it is from the metabolism that is stored in the cell. Second source is aerobic respiration of the cell or aerobic glycolysis. So, it is the ATP plus inorganic phosphate plus energy from carbs, from the carbohydrate, which forms the ATP. These two sources are mostly used when the muscles are in resting condition. During an activity, during any activity, the ATP source is the anaerobic is the anaerobic respiration. Okay. Now, anaerobic respiration means what? Like the muscle glycogen, that is via the blood, breaks down into glucose, right? And then this via cellular respiration breaks down into 36 molecules of ATP and also lactic acid and pyruvic acid. Okay. In very heavy exercise, like when there is like very heavy exercise, the ATP comes from the phosphagen system. What is the phosphagen system? It is the creatine phosphate that breaks down into creatinine plus ATP. That is during that is during exercise. Okay, during exercise, ADP plus creatine phosphate, it breaks down into creatine and ATP. Again, the same thing in resting state follows the opposite system. The creatinine and the ATP in resting state, it will combine together and be stored again as creatine phosphate in the muscle for further use. Okay, these are the various sources of energy. So, one is the stored form that is constantly happening due to the cellular metabolism. Then we have the aerobic respiration during the resting phase, and during the exercise phase, it is the anaerobic and the phosphagen system. Whenever this contraction is happening, there is generation of heat. Contraction and relaxation of skeletal muscle generates heat. Okay, so what is this heat? Heat is formed after the breakdown of ATP from the unused fraction from the unused fraction of ATP. This is how the heat is generated. Now, what are the types of the heat? What are the types of the heat? Types of heat generated in skeletal muscle fiber. Okay, what are the types of the heat that is generated in the skeletal muscle fiber? First one is the initial heat. Okay, initial heat, it is of again two types. One is the activating heat, another is the shortening heat. Okay, now at this initial heat is also a part of the contraction heat. Now again, what are this two type of heat, that is activating heat and the shortening heat? Activating, as we can see from the term activating heat. So obviously this is the heat. It is the heat generated during the contraction of muscle. So for the contraction of muscle, what did we see? There is like binding of the actin and the myosin, right? A very quick revision. So this is like the actin, right? This is say the actin molecules. So when this myosin head, is bound to this actin, okay, and there is like power stroke, the heat generated during the contraction. This is our activating heat. Second is the shortening heat. What is the shortening heat? This is the heat that is produced during the shortening of muscle fiber. Which muscle fiber? Obviously the skeletal muscle fiber. While in contraction. So in context to that, what did we study? If this is the actin. Okay, this is the actin molecule. And this is the myosin. So what did we study? That there is like this power stroke which is at 45 degrees, right? There is like the bending of the myosin head, and at 45 degrees there is that power stroke. So here what happens? The actin molecule is pulled by 11 nanometer. So when the same pulling of this actin molecule happens in many sites, then obviously the length of the skeletal muscle shortens, it reduces. So during that shortening or reducing of the length of the skeletal muscle, the heat that is generated from the fraction of ATP due to actin myosin binding is the shortening heat. Is the shortening heat. Next is the relaxation heat. Next is the relaxation heat. It is the heat produced during unbinding of myosin from actin. Right? After the power stroke is over, the one ATP molecule will bind with the myosin head and it will pull the myosin away from the actin, right? So the actin and myosin will have no contact and that will ensure the relaxation, right? Again, if I have to draw this one for our revision purpose, what it stands out to be after the contraction is over, the myosin head is pulled away, right? The myosin head is pulled away from the actin. Now, when this is happening, one ATP comes and binds, only then there is pulling away of this myosin from the actin. So, this also generates heat. This is called as the relaxation heat. Next one is the recovery heat. It is the heat produced after relaxation and while the muscle goes back to original state. Rather, we can say it is between one relaxation completion, like one relaxation phase is over, and like after one relaxation phase is over and before the next contraction begins. So this contraction and relaxation is like a cycle process. It's a cyclic process. So after the one relaxation is over and the time and during which the contraction has not started. So period after completion of relaxation and before the starting of next contraction is what is the recovery phase. Now during this phase also little bit of heat is produced by the lengthening of the skeletal muscle fiber. During contraction it becomes smaller. Now during relaxation and recovery state it will again enlarge and go back to its original condition. It is seen that little bit of heat is generated during that state, that condition, which is called as the recovery heat. Okay, so these are the various type of the heat that is generated during the contraction and the relaxation phase. Okay, hope this concept is clear. We have studied about the excitation contraction coupling, about the muscle contraction and relaxation, and also the various sources of energy and also the heat produced. Okay, so this is all for our muscle physiology now. Tomorrow we would be starting with our next unit, that is the cardiovascular system. Thank you.